Hello. Today we're going to talk about nervous tissue. So we're going to start, this is part one of nervous tissue. The next video will be part two of nervous tissue. So our nervous system is made up of billions of neurons. And neuroglia, which are support cells for those neurons so they can function properly and our nervous system can function properly. It's divided into two different segments. We have our central nervous system, which is our brain and our spinal cord. And then our peripheral nervous system, which is anything outside of that brain and spinal cord that comes off that brain. Like we have what are known as cranial nerves that come off that brain, or all of the nerves that come off of our spinal cord. Those make up that peripheral nervous system. So as I just said, that central nervous system is the brain and the spinal cord. The brain is housed in that skull. There's about 85 billion neurons that make up your brain. The spinal cord goes from that foramen magnum, and it goes through that central canal or vertebral, that central canal of that vertebral column, and it travels down the entire length of that vertebral column. And this has about 100 million neurons there. Your central nervous system processes your thoughts, emotions, keeps all your memories. While that peripheral nervous system, as I said, it's everything outside of that central nervous system. So all the nerves that come off your brain or your spinal cord, all of those sensory receptors that allow you to feel different sensations. These are part of that peripheral nervous system. So a nerve, it is a bundle of axons. It's usually hundreds to thousands of axons. There's that connective tissue. There's usually blood vessels found within that nerve because they need nutrients. They need that oxygen to function. They need glucose, different ions. They need a way to get rid of their waste products. And they're lying outside the brain and that spinal cord. I mentioned those cranial nerves. We have 12 pairs, a right and a left. These come directly off of the brain. We have 31 pairs of spinal nerves. Test question. Each nerve is going to follow a certain path to go to some specific place in the body. We have sensory receptors. They're not just in your skin, but those touch receptors. We have photoreceptors in our eye. Olfactory receptors. This is what allows us to smell. So they're helping us to detect any kind of change in the environment around us. But we also have other sensory receptors that are detecting the internal environment, like your chemistry makeup of your blood, the different contents going through that digestive system. This is all being monitored. So our peripheral nervous system has a sensory or afferent division. This is going into that central nervous system. So sensory information from your skin, like touch, temperature, pain, proprioceptive. Proprioception is where your joints are in space. I mentioned them before. I think in the muscle chapter I mentioned. But knowing where your joints are in space, what degree of bend you have to your joints, your brain needs to know this information so they can take that information, and when you get up to move, it can create a plan of how to get up and move successfully. Without knowing where your joints are in space, you may not make it all the way up. You may fall over. So knowing where your joints are in space is very important. We also have our special senses. That sense of smell, taste, vision, hearing, and your equilibrium. We will go through the special senses chapter after we get through the different parts of all the nervous system. We'll get through these. We have a motor division or efferent. Efferent goes out. So it is going away from the central nervous system. It is going to effectors like muscles and glands. This motor division can be split up even further. We have that somatic nervous system. Somatic means muscle. So that is information going from the central nervous system to your skeletal muscles. This is your voluntary movement. We also have that autonomic nervous system. This autonomic information is going from that central nervous system to smooth muscle, cardiac muscle, and glands. This one is that involuntary, because again, we can't control how fast something is moving through our digestive tract. We cannot control how fast or slow our heart beats. We're not controlling hormone secretion. Those are coming from glands. Your sweat glands secreting, we're not in control of that. That is why this one is involuntary. And this autonomic nervous system is made up of this sympathetic, which is that fight or flight type of response. That fight or flight, you get really anxious or stressed before you get up in front of a class and give a presentation about something. That's your sympathetic nervous system, that fight or flight, that increased heart rate, that increased sweating, especially in the palms of your hands. Just kind of that overall anxious feeling. That's your sympathetic nervous system. Then we have the parasympathetic, which is that rest and digest. That parasympathetic rest and digest is doing just that. It's helping to digest your food. It speeds up that digestion from, if it slowed it down from that sympathetic, it speeds it back up. The other autonomic nervous system we have is this enteric plexus. This is going directly to the digestive canal. So this is known as that gut brain, or the brain of the gut, is that enteric nervous system. Test question. So our nervous system in general, it lets us smell what is happening. So you can smell what's going on around you, smell your food. Allows you to produce speech. It creates those memories of past events that have happened, allows you to move your body. And it regulates those internal organs. So that sensory function is going to detect some sort of stimuli. Could be internal, could be external. Inside the body, outside the body. It's taken into that brain where we hit this integrative function. This is where we process that sensory information. We analyze it, we make decisions about it, and we figure out appropriate responses. Those responses is some sort of motor function or output. So it takes that sensory information that was taken in, such as it's hot or it's cold. That's sensory information that went into your brain. Your brain processes it and decides what kind of output. If it's hot, it's going to cause you to sweat. If it's cold, it could give you goosebumps or shiver to warm you up. So it'll take that information and it'll send a response to those effectors. It can cause muscles to contract or glands to secrete. Sweat comes from a gland. So those are one of those responses. So this goes through, we have that central nervous system, that brain and that spinal cord. That peripheral nervous system is that cranial nerves. Spinal nerves, it has that enteric plexus. It's going to that digestive system. And it also has those sensory receptors in your skin. So there's a lot of different things that make up this peripheral nervous system. Now we have it on the chart. We're looking into the central nervous system. So our sensory division could be somatic senses, could be special senses, is picking up something. Again, it's hot. That is something we're picking up. Or maybe we smell cookies being baked. Or some other type of food that you like. Well, that goes up into your central nervous system, and then information comes out. It could go to that somatic nervous system to cause your skeletal muscles to do something. Or it could go to that autonomic nervous system. It could go sympathetic or parasympathetic, which will go to the smooth muscle, cardiac muscle, or glands. Or it could go to the enteric plexus, which is smooth muscle and glands of the digestive canal. So just depending on what kind of output we get from that central nervous system is going to depend on the input that it receives. So let's look at the histology of our nervous tissue, because we have to go down to the microscopic level to be able to see what is actually happening. So our nervous tissue is that are neurons and those neuroglia. Neurons, just like skeletal muscle, are electrically excitable. They can respond to different stimuli. They can convert it into that action potential that we first mentioned in the muscle chapter. So a stimulus is any change in the environment that is strong enough to initiate a nerve impulse. COVID is a really good example of this because there's people that lose their sense of taste or smell with COVID. People who've lost their sense of taste or smell, no matter what they smell around them, no matter how strong that stimulus is, they're never going to send that information into their brain. Someone who did not lose their sense of taste or smell, or they got it back post-COVID. You smell something, it hits those sensory receptors in your nose, it has to get to those sensory receptors in your nose to send that nerve impulse. If it's a very faint smell, may not even get up to the sensory receptors in your nose to send it into the brain. And then that nerve impulse or that action potential. This is an electrical signal that's going to travel along that membrane of a neuron. Again, we're going to use ion channels in that membrane. Ions have that positive and that negative charge. So that positive or negative charge is changing that charge of that membrane. It's creating that electrical signal by moving those ions. Once that nerve impulse starts, it travels very fast. Some nerves are very tiny and can send information short distances. Others are very long. Like the nerve that goes down to your big toe starts in your spinal cord. And that is just one nerve that's going all the way down to move that big toe or pick up sensations from that big toe, that hallux. So let's look at what makes up a neuron here. All neurons are going to have a cell body. That cell body is going to contain different organelles, ribosomes. Ribosomes make our proteins. But here's the difference. In a neuron, we have Nissl bodies. Instead of that rough endoplasmic reticulum, it's Nissl bodies that make those proteins. It's just a big cluster of that rough endoplasmic reticulum, but they're called Nissl bodies in a neuron instead that makes those proteins. Task question. We have neurofibrils. These are intermediate filaments. There's microtubules, lipofuscin. These are found in older neurons. Kind of a granules. We don't really know what it does, but it doesn't seem to be pretty harmful to those neurons anyways. There's somatic spines, or these little projections where we have these receptor sites that bind different chemical messengers. And then we have ganglion. A ganglion is a collection of cell bodies outside the central nervous system. Another test question. The nerve fiber are extensions or processes that come off of that cell body. They're either dendrites or axons. A dendrite receives information. So that's the input. An axon sends out information. So that information that came in on that dendrite is going to propagate or travel towards another neuron along that axon, or it could travel towards a muscle fiber or a gland. At the beginning of the axon, we have this axon hillock, which is just a cone-shaped kind of elevation that kind of weeds down into that axon. There's an initial segment. There's a trigger zone, which is the junction of the axon hillock and the initial segment. Axons contain mitochondria. Energy needs to be made. Microtubules and neurofibrils are all found in that axon. The axon terminal. this is at the end. And it's going to divide into many processes that create these synapses. That synapse is that site of communication between one neuron and another neuron, muscle, or gland. The synaptic end bulb is that swelling at the end. When I was drawing it in muscle cells, there we go, that's what I wanted. I was drawing this larger portion that comes out here. This is that synaptic end bulb. Varicosities, which are swellings at that axon terminal. There's synaptic vesicles, those membrane enclosed sacs that have our neurotransmitters in it, which are those chemicals that are being released for that nerve cell to talk to the next nerve cell. Information, they travels pretty quickly. It can travel 200 to 400 millimeters per day. It can go anterograde, which means it's going forwards, traveling down that axon to the next. It can technically go retrograde or backwards, so traveling back up that axon towards that cell body. It does happen. It's rare for information to go backwards, but it is something that does happen. So here's our picture. We have several dendrites. These are all dendrites here at the end. These are receiving sites for all of our information that is coming into that nerve. It's going to travel down this cell body. We have that axon hillock. Oops, that's what I wanted. This axon hillock, which is kind of that cone shape that goes down to that initial segment that goes down to that axon. Whoa, very small. If we get down to the bottom, we have this axon terminal with these synaptic end bulbs. These are what reach out to communicate this nerve's information with the next nerve, gland, muscle. So here we looked at pictures like this when we looked at the tissue chapter in chapter 4. We have these little dendrites coming off. We have that big cell body in the middle. And then we have that axon that's going off to the next nerve, muscle, gland to send out its information. Here's another one. You can see the cell body, the axon that started, that branches off. This is a motor neuron. So this is this neuron that is going to those muscle cells to send out that information to tell these muscles to contract. So our neurons, they are classified based off of their shape and or location. We have multipolar neurons. This is one of the most common because there's several dendrites at one end and one axon at the other. That is this. This is a multipolar neuron. So these are in the brain, the spinal cord, and most of our motor neurons are multipolar. We have bipolar neurons. There's one dendrite and one axon. This is in your eye, your ear, your nose. So these are found more in special senses. Pretty sure that's a test question as well. That allows you to take in this information going in that one dendrite and back out that one axon. Now we have that pseudo-unipolar. Pseudo means false, or it's also known as a unipolar neuron. It has those dendrites. It has one axon, and they're both kind of fused together, and they're continuous with that cell body kind of found outside of that. I've got a picture of it here. Here's that pseudo-unipolar here. So we've got dendrites on one side, axon terminals on the other, and then it's just one big long axon slash dendrite with the cell body off to the side. Here's these bipolar neurons, that cell body with that one main dendrite on the one side, that one main axon on the other. Then these multipolar that we mentioned that has the cell body on one side with the dendrites coming directly off the cell body with that axon on the other side. So let's look at these other neurons. Or these pseudo-unipolar. They're going to be your sensory receptors. So they're going to pick up that touch, pressure, pain, or thermal, like temperature type stimuli. Purkinje cells, they're found in the cerebellum. Test question. These are specific to the cerebellum. Pyramidal cells, they are named because their bodies have a pyramid shape, and they are found in the cerebral cortex of the brain. That is their main location. So these Purkinje cells, you can see all of these dendrites. So there's a lot of information coming into this one cell from all these branches. And then they travel out this one axon to those axon terminals at the end. So it's sending out just as much information that it is receiving. These pyramidal cells over here, as I mentioned, they have that kind of triangular, at least in 2D it's triangular. In 3D it's kind of got that pyramid body shape with lots of dendrites coming in with that one axon coming out. So there are different types of neurons. We have sensory neurons. Again, I mentioned them before. They are afferent. They are going towards the brain. They are picking up information on their dendrites, and then they send that information into the central nervous system. Most of them are that unipolar or that pseudo-unipolar. Same thing, but it's the same type of neuron. Motor. Our motor neurons are efferent. They are taking information away from that central nervous system. They are mostly multipolar. Then we have interneurons. Interneurons are also known as association neurons. Whoops, losing things. They are found usually within that central nervous system. They are found between sensory and motor neurons. This will kind of come into play when we start looking at some later chapters 13 and 14. These interneurons are going to really come into play. Most of these interneurons are also multipolar. So here we have a sensory receptor. This sensory receptor is going to pick up some touch. Something touched this sensory receptor. It's going to take this information into the central nervous system. It will connect to this interneuron, which will process this information, and then it'll send out some sort of response to that touch. It could send out to turn your head to look over your shoulder if somebody touched your back from behind. There's a lot of different responses, but it depending on what type of response or stimulus that was received. We also have those neuroglia. I mentioned that they're support cells for those neurons, but they make up about half of that central nervous system. They're smaller cells. They do not generate or propagate nerve impulses. These cells can also multiply and divide. This is something that neurons really cannot do. They can't really multiply and divide, but these cells can. They work to fill in spaces kind of between these neurons. A glioma is a form of a brain tumor that is formed from these neuroglial cells. They are very highly malignant and they grow very fast. So we're going to go through all of these different types of cells listed here. We have those astrocytes, oligodendrocytes, microglia, ependymal cells. These are only in the central nervous system, while the Schwann cells, satellite cells, they are found only in the peripheral nervous system. So let's go through these. Astrocytes, first one on our list. Your book says they are star-shaped with many processes. We have protoplasmic astrocytes and fibrous astrocytes. Protoplastic are found in gray matter. Fibrous is found mainly in that white matter. They make contact with capillaries. That is their main job. But they can also have them make contact with neurons, pia matter. This is stuff that kind of wraps around the brain. So they support those neurons. They wrap around capillaries because we need to protect that central nervous system from any kind of harmful chemicals, substances. They create that blood-brain barrier, those astrocytes do. Blood does not come directly in contact with the brain, with the neurons of the brain. These astrocytes make sure that there is that space between that brain and those capillaries, so only nutrients that are needed can get through. So in an embryo, this is the first 8 weeks of development, those astrocytes will actually secrete chemicals that regulate the growth, migration, and connection of neurons in the brain. Kind of cool. They help, since they wrap around those blood capillaries, they're maintaining the environment in the brain so these nerve impulses can travel and propagate well. They also are thought to play a role in learning and memory. They help to form new neural synapses. So these astrocytes here, we have these protoplasmic astrocytes on this side. Here's another protoplastic astrocyte. We also have these fibrous astrocytes. You can see how they're coming in contact with these capillaries. This big blood capillary here. Not really big, but they're coming in contact to help kind of work to maintain the environment and only allow what should come through to come through. So here's some neuroglial cells, or those star-shaped astrocytes, in a really cool electron micrograph. Next cell, the oligodendrocyte. An oligodendrocyte is only found in the central nervous system, and they wrap around the axons. As they wrap around these axons, it is called a myelin sheath. That is what they are creating here. This myelin sheath helps to insulate these axons. And by insulating these axons, it is increasing the speed these nerve impulses are sending this information. Microglial cells or microglial-cytes. These function as phagocytes. So they act like as your immune system of the nervous system. So they clean up the area if something happens. Even during normal development, there's cellular debris that can get created, and these microglial cells are going to come in and help to clean it up. They will phagocytize microbes, and they will clean up any damaged nervous tissue. That is their main job. Ependymal cells. Ependymal cells also help to form that blood cerebrospinal fluid barrier. So they line the ventricles of the brain. This is open spaces within the brain and the central canal of the spinal cord. These again are open spaces within that brain and that spinal cord. So they're helping to kind of protect and only allow, again, those certain substances through. If I come back up here, here are those ependymal cells. So this is like a single layer, sheet, like layer going through of all of the different, these ependymal cells that are only allowing certain nutrients to come through. Now we're into the peripheral nervous system, these Schwann cells. These Schwann cells make the myelin sheath for those axons in the peripheral nervous system. The difference, come up here to my picture. Here is an oligodendrocyte. You can see on this pink oligodendrocyte how it has many of these little foot-like processes that are going out and forming this, looks like this myelin sheath. This is in the central nervous system. So this one cell creates several of these pieces of myelination. In the peripheral nervous system, one Schwann cell creates one myelin sheath. It wraps around one axon in one area. A Schwann cell can take up to 20 unmyelinated axons inside of that one cell as well. So here, let's look. Here is a Schwann cell. Here's this axon down here that is myelinated. This Schwann cell just wraps around and around and around and around and around. But this is one cell. This is one cell. This is one cell. This is how Schwann cells are different. Each Schwann cell creates one myelin sheath. Versus this unmyelinated axon over here. This unmyelinated axon will take in several axons. And all it's doing is just kind of protecting those axons. But those axons, those Schwann cells, just keep wrapping up one cell, one cell, one cell to kind of help kind of hold everything together. So this myelination, what is happening in this myelination? So this myelin sheath, it comes in and it wraps around and around and around and around and around and around and around this axon. These both oligodendrocytes and Schwann cells are made of lipid and protein. When an axon is wrapped around and has this myelin sheath, it is known to be myelinated. So it is helping to electrically insulate these axons to help increase the speed of a nerve impulse that is traveling along that neuron. Excuse me. We have those Schwann cells in the peripheral nervous system and those oligodendrocytes in the central nervous system. So I mentioned each Schwann cell wraps a single axon by spiraling around it up to 100 times. A neurolemma. This is only for peripheral nervous system axons. These Schwann cells can help an axon repair and regenerate itself. It only works with those Schwann cells. With Schwann cells, they have this myelin sheath gap. This space between one cell and the next. Another one up here. That is a myelin sheath gap. It's also known as the node of Ranvier. It's a term that still gets used by older docs, older teachers, that this is how I learned it was the node of Ranvier, and they've since changed it to that myelin sheath gap. But they're just those gaps between those Schwann cells. And those oligodendrocytes, they are found in that central nervous system only, and one oligodendrocyte can wrap around several different axons. And the oligodendrocytes cannot help an axon in the central nervous system regenerate. If an axon in that central nervous system gets damaged, it's done. There's no regrowing it back. An unmyelinated axon is just axons without that covering on them. They don't have that myelin sheath, that wrapping, that insulating of this axon here, that this cell over here, all of these axons are technically considered not unmyelinated because they don't have that Schwann cell that wraps around them hundreds of times. So an interesting fact, the amount of myelination increases from birth through maturity. this increases the speed of a nerve impulse conduction. An example, if you've ever played with a baby, a newborn, any child under 1 year of age, you notice it takes longer for them to respond to something. That is because those axons have not been fully myelinated yet. That speeds up that nerve conduction. That is also why they're not that coordinated yet. The responses aren't as quick as even a child or an adult. It usually takes 12 to 18 months after birth for those myelinations to complete that process. So I'd mentioned it before, a ganglion is a cluster of cell bodies in the peripheral nervous system. A nucleus is a cluster of cell bodies in the central nervous system. One of these two is a test question. A nerve is a bundle of axons in the peripheral nervous system. A tract is a bundle of axons in the central nervous system. So this is the process of myelination, how this cell comes in. It's going to start wrapping around, then it starts wrapping around more and more and more and more and more till it's got this myelin sheath in here that's keeping this axon traveling, this information traveling fast. This unmyelinated just has several axons within this one cell. So here you can see the myelinated and the unmyelinated axons in a slide. And so we mentioned gray matter and white matter. So what is gray matter and white matter? White matter is where we're going to find those myelinated axons. There will be some unmyelinated in the white matter, but it's predominantly that myelinated axons because that myelination gives it that white appearance. Gray matter, on the other hand, this is where we've got cell bodies, dendrites, unmyelinated axons, axon terminals, and neuroglial cells. So there's a lot more other types of cells, support cells, in this gray matter. In the spinal cord, white matter is on the outside, gray matter is in the middle, and it's kind of in this butterfly pattern. So our spinal cord kind of has this appearance. The inside kind of has this butterfly appearance to it. This is a poorly drawn spinal cord. In the brain, the gray matter is on the outside, while the white matter is on the inside. So that's a much better drawing than mine. We have the spinal cord here. White matter is on the outside. Gray matter is on the inside. And then on the brain, you can see the gray matter is on the outside. There's a little bit on the inside in certain areas, but that white matter makes up most of the inside. Here is an actual spinal cord and an actual brain, where you can see that white matter, that gray matter. So let's start looking at how neurons send out information. They can be sent out in braided potentials. This is sending information short distances. or they can send out that information in action potentials. We mentioned the action potentials in the muscle chapter, but this is what allows that communication over long distances. We have a membrane potential. This is that difference in charge across that membrane. And then we have that resting membrane potential. This is where a cell sits at rest. So that charge of that cell at rest. Does not say it here. It is usually between negative 70 and negative 90 millivolts. That is where we're going to find that charge inside of that cell. And this is its resting membrane potential. That current is that flow of particles. So that flow of ions that move across that membrane. The electrochemical gradient. This electrochemical gradient, when we have certain ions, sodium, potassium, chloride, phosphate, that are moving across that plasma membrane, they are going down their concentration gradient. This is going back to chapters 2, chapters 3. So ions like to move from that area of high concentration to that area of low concentration. So I have a cell. Outside of my cell, I have sodium in very large numbers. Inside my cell, I have potassium in very high numbers. So potassium's going to leave that cell because it's going from that area of high concentration inside that cell to that area of low concentration outside of that cell. Sodium is going to go into that cell because sodium has a very high concentration outside of the cell, and it's going to move inside of that cell. So positively charged cations move towards negatively charged areas and vice versa. These ion channels open and close because of gates. So we have different types of channels on our cells. We have leak channels. These ones are always open. We have those ligand-gated channels that I mentioned in the muscle section that open and close depending on if something attaches to that channel or not. We have mechanically gated channels. These are like our touch receptors. Are these mechanically gated? Changing the shape of that receptor opens that receptor. So somebody touching you changes the shape of that mechanically gated channel that's causing that channel to open. And then we have voltage gated. These voltage gated channels are going to open and close depending on the charge inside that cell, which I all just explained here. The main thing is with these leaky channels, we have more potassium leaky channels than sodium leaky channels. Potassium is more leaky than sodium. That's like the main difference here. Ligands, mentioned neurotransmitters, but hormones can bind to those ligand-gated channels and even certain ions. I mentioned that mechanically gated, so if a force distorts that channel to open it, and then those voltage gated is going to open by the change in the charge of that cell. So this just kind of goes through that leaky channel versus that chemically ligand gated channel. Something needs to bind to it for that channel to open. Mechanically gated, something needs to change the shape of that channel to open it. And then that membrane, that charge channel. So it opens or closes depending on the charge of that membrane. Nice chart from your textbook that goes through what all of this means. So let's start by looking at our resting membrane potential. That resting membrane potential is where that cell sits at rest. It is created because of the ions, the proteins, the ATP that is found inside of the cell versus outside of the cell. So that membrane creates this separation of positive and negative electrical charges to form. It's usually listed in those millivolts. The greater the difference, the larger the membrane potential is. So inside the cell versus out, we usually have inside the cell usually sits more negative than outside of the cell. Even though we tend to have an equal number of positive and negative charges, that cell usually, just because the proteins usually have more of a negative charge to it, which is helping to keep that membrane where it is. It's typically negative 70 millivolts is that resting membrane potential. At this point, it is said to be polarized. We've got very large numbers of sodium and chloride found outside of the cell. We have potassium, phosphates, ATP, amino acids. Amino acids are used to make proteins. This is found inside of the cell. And then those leaky potassium channels and the number of potassium channels, potassium's diffusing out of the cell, sodium can diffuse down their concentration gradient inside of the cell. That's helping to maintain this resting membrane potential of about negative 70 millivolts. So these potassium leak channels, these sodium leak channels, and then we have a sodium potassium ATPase pump. What this is doing is taking those potassiums that leaked out of the cell and those sodiums that leaked into the cell and pumping them back to where they belong. So pumping sodium back out of the cell, pumping potassium back into the cell where both of them belong. So this resting membrane potential is created because those negative charges unable to leave the cell. Those anions don't freely move. They cannot follow potassium. And then that sodium potassium ATPase pump is helping to maintain that membrane potential by pumping sodium back out and pumping potassium back in. Because of the sodium potassium pump, that negative 70 millivolts can differ by up to approximately 3 millivolts either direction. But it's usually averaged about 70 millivolts. So that's why we tend to keep it at about negative 70 millivolts. So let's
outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode will be about of minus 70 millivolts. The membrane potential depends on two factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, more potassium ions inside the neuron than outside. The membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more stable or leaky to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at minus 90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to minus 70 millivolts. Sodium potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential.
So let's first start by looking at graded potentials. A graded potential creates a very small deviation from that resting membrane potential. It can make the inside more negative, or it can make the inside less negative or more positive. So we have different types of graded potentials. We have hyperpolarizing graded potentials. This hyperpolarizing is making the inside more negative. Depolarizing graded potentials makes that inside less negative or more positive. So as these graded potentials come, can cause a mechanically gated or a ligand gated channel to open or close, and it can make that cell more positive or more negative. This mainly occurs at dendrites. And it also can happen at a cell body because sometimes one neuron can attach to the cell body of another neuron. So this is mainly where we get those graded potentials in. So this graded potential can hyperpolarize and cause that cell to become more negative, or it can depolarize and cause that cell to become less negative or more towards that positive. The difference with a graded potential versus an action potential that we talked about with in the muscle chapter, graded potentials, they can vary in size. They can vary in strength. They can be large, they can be small. It just depends on how many channels they open or close and how long they stay open. These opening and closing of these channels allows these ions to move back one direction or another. For the most part, that stays pretty localized, but it can start to spread to adjacent areas in either direction. But they tend to die out before that information goes too far. So where that graded potential dies out is known as the decremental conduction. It's usually within a few millimeters of where it starts. So if we're going to hyperpolarize, it is caused by that neurotransmitter glycine, which is causing that cell to become more negative. So glycine comes and binds down here at the bottom. It's going to allow negative ions to come in, more negative ions to come into that cell, which is causing that cell to become more negative. And that negative ion that is found outside of that cell is chloride. Graded potentials, even though they tend to die out, they can become stronger. They can last longer. And it's by that summation. So that summation is where the adding together of more and more graded potentials. So if we add two depolarizing graded potentials together, that creates a larger depolarizing. If we have two depolarizing, that's the same thing. Two hyperpolarizing gives us a larger hyperpolarizing graded potential. One depolarizing, one hyperpolarizing, they cancel each other out. So we only have one stimulus that creates a pretty small bump in that resting membrane potential. Two stimuli goes a little bit larger. Three stimuli come in, it gets even larger. And this is where action potentials can kind of come into play. We have, well, that action potential that we mentioned in the muscle chapter. These are sequence of rapid events that occur. It is that nerve impulse that has that depolarizing phase. It'll have a repolarizing phase, a hyperpolarizing phase. But for an action potential to actually take off, for something to happen, it has to reach a threshold. And that threshold is usually about 15 millivolts above that negative 70. So that's about 55, negative 55 millivolts to cause this depolarization where that cell becomes positive. That repolarizing is where we're taking that cell back down to its resting state at about negative 60 or negative 70, millivolts. That hyperpolarization is when that cell becomes more negative than that resting potential. So I think I'm going to go through this and then I'll draw some pictures out. We have a sub-threshold stimulus. A sub-threshold stimulus is a stimulus that is not strong enough to cause that cell to reach threshold. So if we do not reach threshold, we're not going to send out that action potential. So think about if something touches you extremely lightly, you may not feel it. It's because we did not create a large enough impulse to send that out so you know something touched you. A threshold stimulus. That means that we have reached that threshold to cause that action potential to be sent. A supra threshold stimulus, we created a threshold stimulus. It was above that threshold, but that action potential still gets sent out. So once that nerve impulse is generated, it's going to go through the same process every time. So we have our resting membrane potential. We have our threshold stimulus. Something touches you. It creates that little bump in the road. Creates this little bump in the road, but then it goes back. Something touches you a little bit harder. Creates a little bit higher of a bump. But it did not open enough sodium channels to cause that action potential to happen. Something touches you even harder. We've opened up enough of those sodium channels that an action potential is going to take place. So these sodium channels open and we're going to depolarize. Those sodium's going to come into that cell, causing that cell to become more positive. We will repolarize. Potassium is leaving the cell, so that cell is becoming more negative. Then we even go into hyperpolarize, where that cell becomes more negative until it goes back to that resting potential. So this key over here tells us what all these colors mean. Purple is that resting membrane. Blue is where we have the stimulus that comes in until that stimulus is enough to cause that action potential to happen. Then that green is when we have those sodium channels open. That kind of beige is where we have those potassium channels open. Those sodium channels are now inactivating. And then this orange is where we have potassium channels are working on closing. Those sodium channels are going back to their resting position. We have this absolute refractory period listed here and this relative refractory period. So in this absolute refractory period, no matter how strong the stimulus, a new stimulus is that comes in, it's not going to send out an action potential, no matter what. In a relative refractory period, this action potential, if it's a strong enough stimulus, it can stimulate a new action potential. That is what absolute versus relative refractory is. And in action potential, it's an all or none process. So it's either going to occur all of the steps or it doesn't happen at all. So I like your book's example of pushing over a domino in a series. So once you push that first domino, if you kind of lightly tap it, it may wiggle back and forth, but stand straight back up. You have to push it hard enough to push against the fall against that second domino to cause that whole row of dominoes to fall. The stronger you push that first domino, it's still going to proceed the same way. It's still going to cause the next one to fall and the next one and the next one and the next one. That's how an action potential works. You have to push it hard enough to cause it to fall. And then once it falls, no matter how hard you push it, it's going to cause the same exact effect. So I was going to draw a picture, but I think I'm going to go through and do this instead.
Send signals over long distances by generating and propagating action potentials. Most action potentials originate near the axon hillock of the cell body in the initial segment of the axon. It then travels the entire length of the axon. A closer look reveals that during an action potential, voltage-gated channels open and close, altering the permeability of the plasma membrane to sodium and potassium ions. Potential stimulus changes the shape of the voltage-gated sodium channels, causing their activation gates to open. This event marks the beginning of phase 1 of the action potential. The sodium ions diffuse into the axon. The membrane potential becomes less negative. This causes more steady sodium channels to open, and the membrane potential slows to 50 millivolts. At this point, two key events occur. The inactivation gates of voltage-gated sodium channels close, and voltage-gated potassium channels open. These two events mark the beginning of phase 2 of the action potential, known as phase. When potassium ions diffuse out of the axon, the membrane potential becomes negative again. However, the membrane potential continues in the negative direction, so the zone is 70 millivolts. This marks the beginning of phase 2 of the action potential, known as hyperpolarization. During this phase, voltage-gated potassium channels close and all voltage-gated sodium channels are released from inactivation. At the end of this phase, ions move from weak channels only, and the membrane potential returns to the resting state of minus 70 millivolts. The new line is then identify another action potential. Summary. Generation of action potential. Voltage-gated sodium channels close. Voltage-gated potassium channels open. Potassium ions diffuse onto the axon, depolarizing it to a negative value. The membrane potential briefly hyperpolarizes. Voltage-gated potassium channels close, and the membrane returns to the resting state of minus 70 millivolts.
I am going to come through. We'll walk ourselves through this because there are questions on the exam that are asking about what is happening at each stage of this process. I'll post this video again. It's also within the PowerPoint. I'll post the direct link to it into Blackboard for you. But first, we have to reach threshold with a stimulus that comes in. So when that threshold stimulus comes in, we depolarize our cell. By depolarizing it, those sodium channels are opening. Sodium is going to rush into this cell, causing that cell to become positive. Goes from that negative 55, which is that threshold, to positive 30 millivolts. Sodium has that activation gate. If I draw a little sodium channel, it has almost a little like ball and chain that'll come. This is technically that inactivation gate. This here. This inactivation gate will come in and block this channel when that cell has reached a certain positive point, that positive 30 millivolts. This inactivation gate, it remains open during that resting state and in that activated state. It closes when that cell starts to repolarize. So our repolarization, our cell is going to start repolarizing shortly after those sodium gates start to open. Those inactivation gates will close on those sodium channels only, putting sodium channels in that inactivated state. So I've got that open sodium channel, got that inactivation gate. So it is inactivated. With this inactivation gate, nothing can come in through that channel. That threshold level of depolarization opens voltage-gated potassium channels. But they open a lot slower, so it takes a while. That's what allows that cell to get all the way up to that positive 30 before that cell starts to become more negative again. The inactivation gate on those sodium channels and those opening of those voltage-gated potassium channels is what causes that repolarization to occur. Potassium will continue to flow out all the way from that negative 30 to that negative 70. But then again, it takes time for those potassium channels to close. So we have that example, that subthreshold stimulus. Something comes in, causes a couple of those sodium channels to open, but it's not enough. Not enough sodium is able to come in during that time to reach threshold. A threshold stimulus comes in, we're able to send off the nerve impulse. With a suprathreshold stimulus, this is what allows you to tell that somebody touched you really lightly, that threshold stimulus, versus touches you a lot harder, that suprathreshold stimulus. It sends out more action potentials to that suprathreshold stimulus to allow you to notice somebody's touching you harder. So that hyperpolarization. Some of those voltage-gated potassium channels are still open, so potassium is flowing out. This is what causes that after hyperpolarizing or that cell to become more negative. Usually gets down to about negative 90 millivolts. Those potassium channels close. Takes a while, but they close, and that's what allows that membrane to go back to that negative 70 millivolts. So potassium channels do not have that inactivated state. They'll either be open or they will be closed. So I mentioned those refractory periods in that picture above. That refractory period is when another action potential cannot be generated to a normal threshold stimulus. So let's look. Absolute refractory. During absolute refractory, no matter how strong that stimulus is, it cannot initiate a second action potential. Why? Because sodium channels are either open, so they're in that activation state, or that inactivation gate is blocking that sodium channel. If that sodium channel is inactivated with that inactivation gate, they cannot open up. They have to go back to their resting state before they can open up again. That is why this is an absolute refractory, because no matter how strong those sodium channels have not gone back to their resting state, so they cannot open up again. Relative refractory. During this time, a second nerve impulse that is stronger than normal can generate an action potential. Why? We have potassium gates open, but those sodium channels have made it back to their resting state. Once those sodium channels are back to their resting state, that is when a new nerve impulse can be generated. So let's kind of talk through. We mentioned the video showed the channels opening in response to a change in voltage. So let's look at what might happen to a nerve that is using neurotransmitters. So this is set up just like with skeletal muscles. We've got these secretory vesicles inside this first neuron. This second neuron has ligand-gated channels that are closed with an inactivation gate. So this nerve impulse is going to travel down this cell. It does the exact same thing that it does. We have these channels that are going to allow calcium to come in. When calcium comes in, these vesicles are going to bind with that cell membrane and release out their neurotransmitter. That neurotransmitter is going to bind. Whoops, I lost a lot more than I wanted to there. My bad. That's what I wanted, sorry. They're going to bind to these ligands that are going to cause these sodium channels to open. When these sodium channels open, sodium is going to rush into our cell. As the sodium starts to rush into this cell, it's going to cause, actually I'm going to draw a graph over here. So we have that threshold, or we have that resting membrane potential, we have that threshold. So we have that -70 and we have that -55. So during this process, we have sent off enough, opened up enough sodium channels so we've reached threshold. Now we've got a bunch of sodium rushing into that cell. At the same time, we have voltage gated potassium channels. They are starting to open. So now potassium is leaving our cell. As potassium leaves our cell, we are going to go down more negative. But what's also going to happen is these inactivation gates are going to come in and block this sodium channel. So now sodium can't come in anymore, but potassium is leaving. As that potassium leaves, our cell goes down to that -70. And it's even going to drop down below that -70 before these potassium channels close up again. And when we hit that below that 70, -70, these inactivation gates are going to move back out of the way to their resting state. So all of this is happening, the same process, but this time we're using neurotransmitters. These neurotransmitters also have to get released off. For that inactivation gate to come in and close. And then for this sodium channels to go back to its resting state. All of this needs to happen at these different steps. Then these action potentials, the way they propagate, which is the next slide, is they're going to travel down this membrane and send this information down the cell. When one cell communicates with the next cell, they're going to use neurotransmitters. But now the change in the membrane potential, that cell becoming more negative in this area, will cause sodium channels over here to open. Then sodium channels over here to open. Then sodium channels over here to open. Then it causes potassium channels over here to open. Then potassium and potassium here. So the change in that membrane charge is what causes channels down that membrane to open. When one cell communicates with the next, that is where we use neurotransmitters to do that full communication. So that is that propagation of these action potentials. So that propagation, again, it's positive feedback. So when sodium channels open in one area and those sodium ions start to flow in, it causes sodium channels next to it to open. Then sodium channels next to that to open. And that's what causes this action potential to propagate or travel down that cell. And because of that absolute refractory that is happening with those sodium channels that are being blocked off by that inactivation gate, that is what causes action potentials to only travel in one direction. Because it can't cause sodium channels going back the other way to open because they're in that inactivated state. So only sodium channels traveling down that cell in one direction can allow those sodium channels to open. So this picture goes through what I just drew and what that video showed. So this is going through each step. We have that resting state. Sodium channels are closed. Potassium channels are closed. We have our inactivation gate open. We get an action potential that comes in. Change in that membrane charge causes this sodium channel to open. So sodium starts flowing in, causing that depolarization phase. Now, that inactivation gate is going to come in and block that sodium channel while the potassium channel's open, which causes repolarization to happen. So potassium is leaving the cell. Our sodium channels are going to go back to their resting state as this repolarization continues. So anesthetics. These are drugs that can cause a loss of sensation, especially pain. They can be used to block pain, like dental procedures. An epidural or delivery of a newborn. A lot of times these anesthetics are actually used to make this person unconscious so they have no memory of pain or any other sensation. That's why when you wake up from surgeries, if you've ever had one before, you have absolutely no memory of what happened. The surgeries I've had, I remember right before I fell asleep. And then when I wake up, I have no recollection of what happened in between that time period. We have those local anesthetics like lidocaine, Novocaine. They block that nerve transmission of pain to the brain. They prevent sodium channels from opening. So we've talked about these action potentials. We've talked about how they propagate, move along that axon. There's two different ways that this information gets sent out. It can be a continuous conduction, means step-by-step depolarization and repolarization of each area. No picture, I'll draw it. So this continuous conduction, I have a axon here. So this area will depolarize, which will cause the next area, which will cause the next area, and it'll travel down the entire axon. Saltatory conduction. Test question. In saltatory conduction, I have my axon. My axon has myelin sheath with that myelin sheath gap. So what these myelin sheaths do and why it allows this information to travel quicker than an unmyelinated axon. Green looks good. So this area depolarizes. It's going to jump that myelin sheath to the next gap and depolarize there, which will cause it to jump to the next gap. Depolarize here, then jump to the next gap and depolarize here, then jump to the next gap and depolarize here. That is what saltatory conduction does, and it allows it to this information to travel quicker because it's jumping from myelin sheath gap to myelin sheath gap to myelin sheath gap to myelin sheath gap. So it leaps from gap to gap. And this is what allows it to travel faster because on that continuous conduction, it has to depolarize every single area. So each area has to depolarize to allow the next area to depolarize, which allows the next one, 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 instead of jump, 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 jump. And that's what allows that information to travel so much quicker in that saltatory conduction. So what affects that speed of propagation? I just mentioned that. The amount of myelination. The more myelinated, the faster it's going to go. The diameter of that axon. The larger the axon, the faster, because it has a larger surface area, which is causing those more sodium channels to depolarize, which is causing that information to travel quicker. Temperature. Temperature affects the speed of nerve propagation just like anything else. The colder it is, the slower that information is going to be sent. We have different types of nerve fibers. We have type A fibers. These are the largest. They are myelinated. They will travel anywhere from 27 to 290 miles per hour. This information is sending in touch, pressure, that proprioception or your position of joints, temperature, and pain types of sensations. But these are also your motor neurons going to your skeletal muscles. This is what allows your skeletal muscles you to start moving almost right away. We have B fibers. These are smaller. They're myelinated still, and they still do that saltatory conduction, that jumping, but they can only travel up to 34 miles an hour because they're smaller. They also have a longer absolute refractory period. So this is sensory information coming from your viscera, so your chest, abdomen, pelvic area. So this is autonomic motor neurons. C fibers. These are our smallest, and they are unmyelinated. Unmyelinated does that continuous conduction, which makes it slow, 1 to 4 miles an hour. They have the longest absolute refractory period. So this is sending pain, touch, pressure, heat, cold, and pain from your viscera. So this picture is showing that continuous conduction, how it has to depolarize it, each individual area along this axon to send that information versus the saltatory conduction that starts at one myelin sheath gap, jumps to the next myelin sheath gap, jumps to the next myelin sheath gap, which jumps to the next myelin sheath gap, which is allowing that information to travel so much faster. So how does our brain know what we're sending to it? Because it's sending action potentials. It's sending these electrical signals. Well, the frequency of these nerve impulses is going to determine how strong the stimulus is. So a light touch has a low frequency of nerve impulses, while a firmer pressure is going to send action potentials at a lot higher of a frequency. So we use a frequency code in our brain. So the number of sensory neurons that receive this stimulus is, and the number of action potentials that they send out is going to determine the type of touch. So a firm pressure stimulates more neurons than a light touch does. So the more neurons you're stimulating, the amount of action potentials they send out is going to determine how strong or light a sensation may be. So this goes through those graded potentials, those nerve impulses or action potentials, and all the different characteristics for each of these. This is a nice chart from your textbook that goes through all of this information. So let's look at signal transmission at synapses. So it is a synapse, we've mentioned it before, that is that communication between one neuron and another, or between that neuron and that effector cell. We have that presynaptic neuron, that is the information that is sending that signal. That postsynaptic cell or neuron receives the signal. And then we have effector cells. They are going to respond. Like you see your phone on the table, your phone is ringing, and those effector cells are going to help you pick up that phone because it's ringing. So this is kind of how everything gets put together. So our types of synapses. We have axodendritic. The nice thing is, kind of like with joints, these tell you where it starts and where it ends. So from that axon to that dendrite. Axosomatic, axon to cell body. Axoaxonal, from an axon to an axon. One of these is a test question. So here's our picture. We have this axon here that is connecting to a dendrite on the other side, creating that axodendritic synapse. Here we have that axon in contact with the cell body. That gives us our axosomatic. Soma is another name for cell body. Then we have the last type. We have an axon come in contact with another axon. That is giving us that axoaxonal type of synapse. Those are those three different types. So in an electrical synapse, those action potentials are going to conduct directly between the plasma membrane through gap junctions. So we're going to go to those adjacent neurons from one neuron to the next through a gap junction. They contain those little tunnels or connexins. That's going to connect the cytosol from one cell to the cytosol of another cell that allows that action potential to travel from that one cell to the next because of the change of that charge of that membrane of that first cell helps to change the charge of the membrane of the next cell. This electrical synapses have a lot faster communication because that information goes through gap junctions, which is what allows for synchronization. So a group of neurons or muscle fibers can fire in unison because of those gap junctions, those little tunnels between those one cell to the next. In a chemical synapse, that was the electrical synapse. This one was the video I showed. So that video. This, a chemical synapse, was my drawing that I drew. So in a chemical synapse, that presynaptic and postsynaptic neuron do not touch. So that picture I drew, that first neuron and that second neuron, they do not touch. They are separated by what is called that synaptic cleft, which is that space between. It is filled with interstitial fluid, which means when that nerve impulse travels down that first axon, it releases out that neurotransmitter. It has to diffuse through that interstitial fluid and bind to those receptors on the plasma membrane of that postsynaptic membrane. That postsynaptic potential is a chemical signal that produces a graded potential. The presynaptic neuron converts that electrical signal, so this electrical signal that is traveling down this first neuron here, this information is electrical, it gets converted to chemical. Because since these two neurons are not in contact with each other, there's this gap between them. They have to use chemicals, a neurotransmitter, to communicate, which creates a synaptic delay because it requires time for that neurotransmitter to travel across that synaptic cleft and start that action potential on that next cell. Most chemical synapses have a one-way transfer of information. So information only goes one direction in that chemical synapse. So this goes through all the steps of that chemical synapse that I drew out on that previous slide. Everything that was happening here in this chemical synapse is written out right here for you. And this picture shows it. So that nerve impulse coming down causes calcium to come into that presynaptic neuron. As more calcium comes in, it causes those synaptic vesicles to fuse with that membrane, which releases out that neurotransmitter. That neurotransmitter binds to that ligand-gated sodium channel that causes that sodium channel to open, which causes sodium to come in. As sodium comes in, that cell becomes more positive. Those ligands get released, which causes that sodium channel to close while potassium channels open, which causes potassium to leave that cell, which causes that cell to repolarize to become more negative again. So when we're looking at neurotransmitters, some neurotransmitters can be excitatory. They can cause that cell to depolarize. They can also be inhibitory, meaning they cause that cell to hyperpolarize. So we have different kinds. We have excitatory postsynaptic potential, EPSP. More test questions between this one and the next one. It's excitatory, so it's going to cause that cell to depolarize. That neurotransmitter that's being released will cause it to depolarization or depolarize. We have the inhibitory postsynaptic potential, IPSP. This is causing a hyperpolarization. So that cell is becoming more negative. So now let's look at this first part. A neurotransmitter that causes a depolarization is excitatory. A neurotransmitter that causes that hyperpolarization is inhibitory. So, as that cell becomes more negative, draw a graph here, that resting membrane potential, an inhibitory signal comes in, it causes that cell to become more negative. It makes it a lot harder, or as this says, more difficult to create an action potential because it's more negative inside of that cell. And we have that threshold that we have to reach for that action potential to be generated. So when that cell becomes even more negative, to be able to reach that threshold takes a lot larger of a stimulus to cause an action potential to happen. So we have those neurotransmitter receptors. So each neurotransmitter can technically bind one or more neurotransmitters. So when that correct neurotransmitter binds, it can cause an excitatory or an inhibitory response. That can be either ionotropic or metabotropic types of receptors that these neurotransmitters will bind to. So an ionotropic receptor. It's a neurotransmitter receptor that has a binding site and an ion channel as part of the same protein. So it's those ligand-gated channels that I've been drawing. If that neurotransmitter doesn't bind, that ion channel remains closed. So we have those binding sites. Stop. Those binding sites here for that channel. We have that binding site with that channel closed to remain closed there. It remains closed until that neurotransmitter binds. So let's draw this again. Now I have a neurotransmitter that is bound, which is going to open this channel. So this all is on the same, what it's saying is this whole thing is one channel. That ligand binds, the channel opens, it's all on the same protein. It'll cause that excitatory or inhibitory response. So that excitatory will depolarize, that inhibitory will hyperpolarize. When we depolarize, we're letting in positive ions. When we inhibit and we hyperpolarize, we're letting in negative ions. A metabotropic. This one we will see more when we get to special senses. But what a metabotropic receptor is down here at the bottom is a neurotransmitter is going to come in and bind. Got this neurotransmitter here that came in. It's going to bind to this protein, to this specific receptor here. When that binds, we have this G protein that is attached to this receptor that is going to go over to this channel in another area and cause this channel to open. That is what it is doing. So a metabotropic receptor that neurotransmitter is going to bind to that receptor, which is going to cause a G protein to travel as a second messenger over to an ion channel to open or close that ion channel. They are two separate parts. They are different components. They are different proteins in a metabotropic receptor. We will see this again when we look at the eye. We will see this again when we look at I think it's the ear is the other one that has metabotropic receptors. So this just shows the ionotropic receptor that is going to cause that excitatory. So that cell becomes more positive. GABA is a neurotransmitter that causes an inhibitory response. So it causes chlorine to come in and that channel to become more negative, or that cell to become more negative. But here's the interesting part. In different areas, like in the spinal cord versus the brain versus going to muscles, cell glands, organs, they're the same neurotransmitter can have a different response. So one that is a good example is acetylcholine, that ACH. Acetylcholine at skeletal muscles is excitatory. It's going to cause that skeletal muscle to contract. But acetylcholine, as it shows here, how it causes that excitatory at skeletal muscles, it can inhibit some of those metabotropic receptors. So it causes that cell to become more negative. Different neurotransmitters are going to cause different responses in different areas. That is where some of this will kind of come into play, where one is more positive, one is the same neurotransmitter can cause different responses at different areas. So how do we get rid of neurotransmitters? It was mentioned in the video that we watched in the muscle chapter, but here's where we kind of go through it. That neurotransmitter can be released from that protein, that channel, and it can diffuse away from that synaptic cleft. So it's just no longer, it's out of reach. So it can't bind to another channel. We can have enzymes come in and break it down. Like that example, that acetylcholinesterase. This comes in and breaks acetylcholine down so that it can no longer bind. It can also be reuptaked by some of our cells. So that cell that originally released that neurotransmitter can take it back up. So it'll take it back up into that cell and they'll put things back together and rebind it back into those secretory vesicles. Test question. So let's look at spatial and temporal summation of postsynaptic potentials. So we talked about graded potentials and how these graded potentials can add up. Well, we have a summation of these graded potentials where they're all kind of added together. We can add up different types of graded potentials, some inhibitory, some excitatory. We can take them and add them up to see what we get. So we have spatial summation. Spatial summation is in response to stimuli that occurs at different locations in the membrane at the same time. So it kind of results from different neurotransmitters being released to that same cell in different areas. Well, we have these temporal summation. Is this response to stimuli that occurs at the same location. So the buildup of different neurotransmitters released by a single presynaptic end bulb and 2 or more times in rapid succession. So let's look. So this spatial, we have these 2 separate neurons here that are coming together to this one postsynaptic neuron. So depending on what they send, that could generate an action potential versus this one neuron connected to this one neuron. That's temporal. So this one neuron is sending these action potentials, and they have to send enough of these action potentials together quick enough to cause, or enough of these graded potentials together to generate that action potential. That's what I should say. Same with the spatial summation. They have to send out enough of those graded potentials to reach threshold to cause that action potential. That temporal is the same way. They have to send out enough of those graded potentials, positive graded potentials, to reach threshold. So, as I mentioned before, they could be excitatory and they can be inhibitory. So, if the total excitatory effects are greater than the inhibitory, but less than the threshold, that excitatory doesn't reach threshold, so that action potential technically does not even get sent out. If the inhibitory effects are greater than the excitatory, that membrane will hyperpolarize, resulting in that inhibitory, that inability to send that action potential. But then we have that nerve impulse. So if the total excitatory effects are greater than the inhibitory, and we're able to reach threshold, then those nerve impulses will be triggered, will be generated. So let's look. Here, we've got excitatory, 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 inhibitory, inhibitory. So we have more excitatory than inhibitory. So some of these are going to cancel themselves out. The inhibitory, excitatory, inhibitory, excitatory. So this is our one excitatory. If it's enough to generate that nerve impulse, it'll send out that action potential. So this goes back to the summary of a neuron, all the different parts of that neuron, and all of their different functions that go into that excitatory, that inhibitory, what type of gates may be there, how these neurons work. They're good to kind of go back and review. We're going to look at some of the different types of neurotransmitters. We have neurosecretory cells, which are neurons that can secrete out hormones. We have different types of neurotransmitters. We have small molecule neurotransmitters, and we have neuropeptides. So our small molecule neurotransmitters. We know the most about acetylcholine because it is released at that neuromuscular junction between the nerve and the muscle, and it creates that excitatory response. It also creates that inhibitory response at those metabotropic receptors. Like it can slow the heart rate when it's inhibitory. There's also amino acids that can work as neurotransmitters in the central nervous system specifically. So let's look at some of these. Glutamate and aspartate are excitatory. That gamma aminobutyric acid, or GABA, and glycine are inhibitory. So GABA is going to open chloride channels because chloride is negative, causes that cell to become more negative, so it's going to hyperpolarize. About half of the inhibitory synapses in the spinal cord are going to use glycine. The rest will use GABA. Test question. So we've got that acetylcholine. We've got these different amino acids. We've got biogenic amines, which are amino acids which have been modified. So epinephrine, norepinephrine, dopamine, serotonin, these are biogenic amines. Norepinephrine wakes you from a deep sleep, released when you're dreaming. It also regulates your mood. Norepinephrine and epinephrine also serve as hormones that are also known as catecholamines, and they come from that amino acid known as tyrosine. Dopamine. This is active during emotional responses, addictive behaviors. Those of you who are addicted to caffeine, if you don't drink caffeine, you get headaches. That's an addictive behavior. Pleasurable experiences. They help to regulate your skeletal muscle tone. They also help with movement of some skeletal muscles. Parkinson's is a disease because of the degeneration of of neurons that release dopamine. Another one, serotonin. Sensory perception, temperature regulation. Helps to control your mood, appetite, and helps to induce sleep. So there's some of these neurotransmitters that cause a lot of different responses here. ATP can work as a neurotransmitter, and there's other purines. So ATP can be an excitatory neurotransmitter. There's different synaptic vesicles that are going to contain ATP. In the peripheral nervous system, ATP and norepinephrine get released together. How about nitric oxide? It is an excitatory neurotransmitter in the brain, spinal cord, adrenal glands, and the nerves in the penis. Nitric oxide synthase makes nitric oxide from arginine, another amino acid. We do not store nitric oxide. We make it on demand because it is a very highly reactive free radical. So it is broken down within 10 seconds of being released because it can cause damage to our cells if it is in our system for too long. Carbon monoxide. Here's another one that people kind of look at and say, what? We make it as needed. It is an excitatory neurotransmitter, and it's thought to protect against excessive neuronal activity. It helps to dilate blood vessels, memory, sense of smell, vision, thermal regulation, so your body temperature, insulin release, and anti-inflammatory activity. Again, we make it on demand, and it gets broken down pretty quickly because carbon monoxide is, in large amounts, is pretty toxic to the body. Neuropeptides. These are neurotransmitters that have chains of amino acids, 3 to 40 amino acids bound together. They work mostly at metabotropic receptors. And again, they're going to have excitatory or inhibitory effects. Some of these are like enkephalins. These are pain relieving that is stronger than morphine. Endorphins and dynorphins. These are opioid peptides. These are our body's natural painkillers. Again, our bodies do make our own opioids, but our body uses them to help relieve some of our own pain. Acupuncture. This is from your book. I thought it was interesting because I also do acupuncture. So it's thought to cause this analgesic effect by helping to release opioids. These endorphins and dynorphins have been linked to improving memory and learning, feeling of pleasure or euphoria, helping to control your body temperature, regulate your hormones that help that onset of puberty, your sex drive, reproduction, but also can play a role in mental illnesses such as depression and schizophrenia. Another one, substance P. Substance P transmits pain. So substance P is a substance that allows you to feel pain. These enkephalins and endorphins suppress substance P to help decrease the number of nerves that are sending out those pain signals to the brain. So substance P and those endorphins and enkephalins have those opposite effects. So substance P can counter the effects of nerve damaging chemicals. So here's a nice chart that goes through a lot of these different neuropeptides. We didn't go through all of them, but there's several. Here, like cholecystokinin is supposed to help regulate and help send out that stop eating signal. Neuropeptide Y stimulates food intake and stress response. Angiotensin 2 stimulates thirst. So there's a lot of different neurotransmitters that are going to do a lot of different things. So let's look at some different neural circuits. A circuit, that neural circuit, is all the different neurons and how they function. So a simple circuit, I thought I had a picture of it, but I don't, is where one neuron stimulates one neuron that stimulates one neuron and so on. So that one neuron is going to go out, stimulate another neuron, stimulates another neuron, and so on. So it's just one neuron that connects to one neuron that connects to one neuron. A diverging, that's not what I wanted. I wanted the highlighter. That diverging circuit, that is one neuron that synapses to several postsynaptic neurons. So that's that divergence. So this is that diverging circuit. One neuron goes to two, or it could go to many, and then those neurons go to even more neurons. So that convergence is that coming together. So that converging circuit is that one neuron, or that several neurons, I should say, several neurons that come together to a single neuron. So here's that one, that converging circuit. These several inputs come down to that one output. That is a converging. Converging comes together. Diverging dives out. We have reverberating circuits. So we have incoming stimulus stimulates the first neuron, which stimulates the second, which stimulates the third, and so on. But here's the thing. Here's a reverberating circuit. So this one neuron stimulates the next, which stimulates the next, but these neurons are sending this information back up, which sends this information back up, which gives us to that output, which is going to send that information back up. Why is that important? Breathing. So that information is going down to breathe in. That information is being sent back up that we breathed in. Okay, now we got to breathe out. That information just is getting sent down to breathe out while that information is getting sent back up to say, okay, we're breathing out. Now we need to breathe back in. Coordinated muscle activities like left foot, right foot, left foot, right foot. We stepped on it with our left foot. So now that information needs to go back up. Okay, now it's time for our right foot. Now we stepped with our right foot. That information needs to go back up so we can step with our left foot. Waking up in the morning and your short-term memory plays a role in these reverberating circuits. Parallel after discharge circuit. So one presynaptic cell stimulates a group of neurons, which stimulates different cells. So let's look. So we have this one neuron that is sending this information out one way, out another way, and down the middle. That out one way is going through a couple neurons. This one's going through a few neurons to come to one output. That's that parallel after discharge. This is how we solve math problems. Is this parallel after discharge? So it's sending this one neuron with this one math problem is sending one part of this math problem out one way, another part of this math problem out another way, while it's sending that information down the middle so they can put all of this information together to come out with an answer, that one output at the end. So let's look at repair and regeneration of nervous tissue. Plasticity. Oh, PS. Test questions. These neural circuits. Is the capability to change based on experience, that plasticity. So these changes could be sprouting of new dendrites, new proteins, changes in synaptic contact with other neurons. Test question. Regeneration. Is that capacity to replicate or repair. Dendrites and myelinated axons may be repaired if the cell body is intact. And if Schwann cells that produce that myelination remain active. In the central nervous system, there is no repair to damage. Even if that cell body stays intact in that central nervous system, they cannot be repaired and regrow. This only happens at peripheral nervous system. So, neurogenesis is that, keep doing that today. It's that regeneration of new, or that birth of new neurons. We have a hormone called epidermal growth factor. This hormone, not only does it stimulate the skin to grow, but it's a hormone that stimulates neurogenesis, new neurons. It can trigger mitosis in non-neuronal cells. Neurons do not go through mitosis. It can promote wound healing and tissue regeneration. New neurons can be formed when you learn new material. So there is a complete lack of neurogenesis in the brain and the spinal cord. Why? Those neuroglia are going to secrete out inhibitory types of chemicals that prevents that regrowth. Those oligodendrocytes do too, which creates the myelination of the neurons in the central nervous system only. There is no growth stimulating factor that was present during fetal development. Once you're born, that goes away. So those oligodendrocytes inhibit regeneration of neurons. Astrocytes will proliferate and they'll form scar tissue. And that kind of acts as a physical barrier to prevent regeneration. So how do these axons repair themselves in the peripheral nervous system? They can only undergo repair if the cell body is intact, if the Schwann cells are functional, and if scar tissue does not form too rapidly. These are things that can prevent those neurons or those axons from regrowing. So let's look at what happens. We have chromatolysis. You will see this word again. Lysis means to break down. You will see this in anatomy 2 a lot, but seeing it now, knowing lysis means to break down, it'll help. So after that injury, Nissl bodies break up into these fine granules. That axon that is distal to the damage will break apart into fragments. The myelin sheath will also deteriorate. This is known as Wallerian degeneration. Test question. Anything past the damage will degenerate. So here's a normal neuron. This is the chromatolysis and that Wallerian degeneration. So we have damage to this axon here. Everything past that damage degenerates. Macrophages are going to come up and clean up the debris. We have RNA and protein synthesis that's going to accelerate, which will cause rebuilding or regeneration of that axon. Schwann cells will go through mitosis, and they will form a regeneration tube across that injured area, so that tube can help guide the growth of that new axon. Axons cannot grow if the gap at the site of the injury is too large, or if the gap becomes filled with collagen fibers forming that scar tissue. So the last part, this regeneration. These Schwann cells have made a regeneration tube here to allow this axon to fully regrow, and it helps to guide the growth of that new axon. So during those first few days, we get buds of regenerating axons, and they begin to invade that tube made by those Schwann cells. Axons can grow at about 1.5 millimeters per day. And they can find their way into that distal regeneration tube. So sensory and motor connections can be reestablished, and some functions can be restored. Schwann cells will form new myelination sheaths. But it takes time, considering 1.5 millimeters per day, depending on how far down that damage is, it could take months before that nerve has regenerated and grown far enough to allow to reconnect and reestablish its functions. It could take months, because 1.5 millimeters a day is very little. So let's go through some different conditions that affect the nervous system as a whole, or stuff that we've talked about today anyways. So, multiple sclerosis. Multiple sclerosis, it's an autoimmune progressive destruction. And what is being destroyed? The myelin sheath in the central nervous system specifically. It usually shows up between the ages of 20 and 40, and it affects females more than it does males, more common in whites. As I mentioned, it's autoimmune, which means your own immune system is attacking those myelin sheaths. And as that myelin sheath deteriorates, it starts to become sclerosis or can form these hardened scars or plaques. There's been lots of MRI studies done on multiple sclerosis, and you can see lots of different plaques on an MRI in the white matter of the brain and spinal cord. So that destruction of that myelin sheath is going to slow down and eventually cause that propagation of nerve impulses to short circuit. So it's going to slow down because that myelin sheath speeds up that information. Getting rid of that myelin sheath slows everything down. People who have multiple sclerosis kind of go through this relapsing remitting, where they have symptoms, it kind of just goes away for a while, then the symptoms come back, then they'll go away, then they'll come back, then they'll go away. Since it usually appears early adulthood in their early 20s, the first symptom usually is heaviness or weakness in the muscles, abnormal sensations, or double vision. So they have that remission period where those symptoms will temporarily disappear. One attack follows another for many years. Usually every year or two, they get some sort of attacks. And it's progressive. So they have this progressive loss. We don't know what causes it. Could be genetics. Certain environmental factors seem to contribute to this. There is a lot of really good new medications out there lately. So they've been able to use this injection of beta interferons, which can lengthen the time between these relapses. It also decreases the severity of these relapses, so it slows the formation of new lesions from happening. Epilepsy. Epilepsy is short, recurrent attacks of motor, sensory, or psychological malfunction. The interesting thing is epilepsy almost never affects somebody's intelligence. It affects about 1% of the population worldwide, and it's an abnormal synchronous electrical discharge from millions of neurons in the brain. Could be abnormal reverberating circuits even. We don't know for sure. But that discharge can send these nerve impulses over these conduction pathways. They can result in people seeing lights that aren't there because they're, depending on where these neurons are firing off from, they're firing off without being triggered. They just randomly fire. It can cause you to see lights that aren't there, to hear noises that aren't there, to have smells that are not there. It can cause skeletal muscles to contract involuntarily. So they've changed the name of seizures since I first learned about them. So now we have partial seizures that start in a pretty small area of the brain, and they produce some pretty mild responses. We have generalized seizures. This covers a larger area of the brain. Usually on both sides, these people tend to lose consciousness. can be because of brain damage during birth, metabolic disturbances, infections, toxins, vascular disturbances, so blood supply, head injuries, tumors, brain abscesses. If fevers get too high in children under the age of 2, it can trigger seizures. Most seizures have no demonstrable cause. There are anti-epileptic drugs out there that can completely eliminate or alleviate a lot of these different symptoms. There's different implantation devices that can stimulate the vagus nerve, which is one of the cranial nerves that we'll talk about, I think in chapter 14. And then sometimes they can go do surgical interventions as a last resort. Excitotoxicity. Excitotoxicity is where there's very high levels of glutamate, which is an amino acid that's in that interstitial fluid of the brain. And it's going to cause these neurons to degenerate because glutamate is a neurotransmitter within the brain, and having excessive levels of it causes this prolonged activation of those neurons to where they pretty much stimulate themselves to death. Oxygen deprivation is the most common cause. So, not enough blood getting to a certain area of the brain means there's not enough oxygen coming. It happens with a stroke. A stroke is where a blood clot gets into that brain and blocks blood supply, which means it's blocking oxygen and nutrients. With that lack of oxygen, that glutamate transporters will fail, which causes that glutamate to accumulate, causing those neurons to stimulate themselves to death because, again, that glutamate works as a neurotransmitter. Some other things. Depression. Depression is a disorder that affects a lot of people in the US, 18 million people a year. These people have feelings of sad helplessness. They have a lack of interest in most activities that they once enjoyed. Sometimes they can even experience these suicidal thoughts. Somebody who is diagnosed with major depression, they have these feelings, symptoms of depression that last for more than 2 weeks. Dysthymia. This is episodes of depression that alternate with periods of feeling normal. So they kind of go that back and forth. Bipolar disorder, also known as manic depressive disorder. So they get like these recurrent episodes of depression and this extreme mania. So they could be really depressed, and then they can go to this extreme elation or mania where they can be awake for days on end. They're constantly going. They're talking 100 miles a minute all the time. And then we have seasonal affective disorder, or SAD. This happens usually in the winter months. People experience depression. It's when those days are short. A lot of people, because lack of daylight, a lack of vitamin D, a lack of sunshine, can cause this seasonal affective disorder. So there's some research out there that shows that depression is actually linked to an imbalance in serotonin, norepinephrine, and dopamine. Heredity is going to play a role, so your genetics. Stress, chronic illnesses. A lot of times people with chronic illnesses start to experience depression because they don't see a way out. A lot of chronic illnesses, they're on medication for the rest of their life. They don't see a way out. Low self-esteem. Hormonal changes can lead to depression. So some of the types of drugs that they use is selective serotonin reuptake inhibitors, or SSRIs. And it can provide some relief for depression because it inhibits that reuptake of serotonin. So it allows it to stay in that synaptic cleft as that neurotransmitter for longer so it can have longer effects. And this was meant to be a 2-part lecture, and I just kept talking, and we have made it to the end of our nervous tissue. Thank you very much for listening, and have a wonderful day.